Well, good morning, everyone, and thank you for the opportunity to speak today. I'm a postdoc in Stuart Ainsworth's lab up in Liverpool, and today I'm going to describe the design of a new consensus toxin that we've generated in our lab. Oh, there we go. So firstly, what is a consensus toxin? Well, these are recombinantly expressed proteins that are designed to represent an average sequence and structure of many native toxins within one toxin family. Although they're still emerging in the toxinology field, they've been evidenced so far for spider, scorpion, and snakebite envenomation. And this has primarily been as a tool for generating broadly neutralizing antibodies. So the first instance of this was by De La Rosa back in 2018, 2019, who used a consensus short chain alpha neurotoxin to generate broadly neutralizing antisera in a horse model. And then, as I mentioned yesterday, they're more commonly now used in phage display systems for monoclonal antibody discovery. However, to date, the primary focus has been on three-finger toxins, and there's been no publications about consensus toxin design for other snake venom toxins, which is where my interests lie. And so what we tried to do was to apply the the principle of consensus toxins to snake venom metalloproteinases. Now, I've not got a great deal of time to talk about SVMPs in any real depth, but they're a primary contributor to some of the hemorrhagic pathology we see in some snakebite envenomings, particularly those by vipers. SVMPs are a diverse range of zinc-dependent endoproteolytic toxin enzymes, and they can be classified by their domain structure into P1, P2, and P3 SVMPs. Now, P1s are the most simplistic. They possess only a metalloproteinase domain, and this happens to be where the zinc binding domain is located, whereas the P2 and P3 SVMPs are a bit more complex and contain these non-enzymatic accessory domains. Whilst P1s are considered less hemorrhagic than their more complex counterparts, all SVMPs possess this metalloproteinase domain, and so we felt that the design of a consensus P1 was probably a good starting point for the project. So with our sequence, it was designed from over 100 different individual metalloproteinase sequences, and that covered 42 different species. As well as the metalloproteinase, it contained a prodomain and then various tags for our downstream applications. Initially, we were quite skeptical as to whether bacterial expression would be fit for purpose, but we decided to give this a go anyway before considering more complex expression systems. And so all the cloning was performed and codon optimized for E. coli expression. And we did this using a cell line called Origami2, which we've had good experience with in the past for producing heavily disulfide bonded proteins. We used an autoinduction media with a dual temperature protocol, and then we performed various solubility tests before purifying via the protein's his tag. We then went on to characterize what we had expressed, particularly looking at antivenom recognition and whether there was any functional activity. And so first, I'll just talk about our expression and our purification. As you can see on this anti-His Western blot, there's a clear upregulation of a protein band between 25 and 35 kilodaltons by the end of our 30-hour expression. And when we looked at the solubility, you can see in the supernatant on this Kumasi stain that there was a significant amount of soluble protein that corresponded to the His positive bands. This is just one representative image of a 200 ml expression that I purified using a His trap column. And as you can see in the fractions, there are three prominent bands, and we suspect these correlate to the immature pre-processed SVMP at about 55 kilodaltons, and then between 25 and 35 kilodaltons, we believe these to be the prodomain and the met domain. With regards to yield, for each of the expressions I've done so far, this is correlated to between 89 to 135 mg per litre. So we're able to generate quite substantial soluble amounts of this protein. Now, although those bands correlated to roughly the right size we were expecting for our consensus, we were yet to know whether it could be recognised as an SVMP. And so what I did was take fractions that I'd shown on the previous slide, and I did a Western blot with same polyvalent antivenom. And as you can see, there's clear recognition of both the pre-processed and the met domain by an antivenom, which gives us confidence that our recombinantly expressed consensus toxin could be recognized by an antivenom that was generated by crude venom. And so we were obviously quite excited by that, as it sort of indicated we were on the right track. And so what we then wanted to do was fully characterize the consensus that we'd made, and it was helpful to be able to compare it back to a number of native P1s that were purified from Echis, Vitis, and Bothrop species. So this is an ELISA experiment where we probed the consensus and the free native toxins with a panel of 13 different Viper antivenoms that covered a broad geographical range and broad genera. As you can see, there was quite a broad recognition of the consensus by the antivenoms, and this was recognised to quite a high dilution. Often the consensus was better recognised than the native toxins, except for perhaps the genus-specific antivenoms. And there were particularly strong responses by antivenoms for Bothrops, the horned viper, for boomslang, and for Echis. And we believe that this indicates our consensus P1 is broadly geographically representative, although antivenom responses by some of the Asian antivenoms were slightly less impressive than other parts of the world. And so for those of you who prefer a Western blot, because we all prefer one technique or the other, I also did a small number of complementary Westerns, again using the consensus toxin and the free native ones, and probed them with antivenoms to cover Vitis, Bothrops, and Echis. And so as you can see, there is again a strong recognition of the consensus P1 by the antivenoms, so that confirms the data we saw in the ELISA. But it also allows us to see that different bands within the consensus sample were recognised to different extents by the antivenoms. So that was a nice additional finding that the ELISA didn't allow us to see. And it also allowed us to see that Vitis and Echis venom produce fewer cross-recognizing antibodies. They tend to have only recognised the consensus or their own genus, whereas the Bothrop-specific antivenom recognised all three of the native toxins, albeit to varying degrees. So now we knew that we had a consensus P1 that was well recognised by antivenom. We next wanted to see if it was functional. And so the next 3 slides are going to show a series of different proteolytic activities. The first one is insulin B. And we digested an insulin B chain with the consensus P1. And then reverse phase HPLC allowed us to evidence that there was a single cut site by the generation of these 2 cleaved products. And coincidentally, that was an identical pattern to what had been witnessed from a P1 purified from Echis leucogaster. That was a really interesting finding. We were then able to use LCMS to obtain masses for the 2 cleaved products, which allowed us to determine that the cleavage site had occurred between the alanine and leucine, as you can see on the insulin B sequence. Next, we wanted to see how the consensus interacted with different components of the blood clotting cascade. And so fibrinogen and prothrombin were incubated with or without the consensus toxin, and then the subsequent samples were run on a gel. And as you can see, the consensus was able to degrade alpha fibrinogen completely, and it had a partial effect on beta fibrinogen, but it was not able to convert prothrombin into thrombin. And then finally, just to describe a kinetic fluorogenic assay, this is one that's used quite commonly to screen drugs against SVMP-rich venoms, and it involves incubating a set amount of toxin or venom, pre-incubated with or without a treatment, and then assessing the ability of the toxin or venom on a synthetic substrate. So as you can see, there was a modest activity from the consensus, particularly if you compare it to the Echis Romani positive control in purple. But we were pleased to see that this modest activity was able to be inhibited by the matrix metalloproteinase inhibitor marimastat. And so the last point I sort of want to touch on is what the keen-eyed and eared amongst you will have noticed. I am persistently showing 3 bands in my consensus sample, and this is despite histag purification. And as I've mentioned, we believe these correlate to the immature form, the met domain only, and the pro domain only. Now, the problem with this is that the pro-domain does not have a purification tag, so it shouldn't really be being retained to be evident on my gel, but it persistently is there in a roughly 1-to-1 ratio with the met domain. And so we can only conclude that despite it clearly undergoing autocleavage, it's somehow remaining attached in a complex, and that's persisting despite much harsher separation techniques that we've tried so far. It's not a completely unfamiliar problem. In the recombinant expression of human ADAM33, it was evidenced, and much more recently for SVMP recombinant expression of native toxin sequences. And so whilst I'd love some input on ways that we might obtain this elusive pure met domain, I guess at this point, does it matter? And that is really dependent on what we're going to be doing with the protein downstream. And so just to summarize what I've described here and talk a little bit about our future steps, I've described the design and high yield expression of a functional soluble consensus P1 SVMP that was well recognized by different antivenoms from a broad geographical range and from broad genera. I was able to demonstrate the specific proteolytic activity that the toxin has. And our next steps now are to take this into horse immunization experiments and try and generate broadly neutralizing SVMP antibodies. Alongside that, we are also going to be doing some map discovery using phage display. And sort of in the background, I am continuing to tinker with trying to isolate that met domain. I've tried a couple of these steps already, and removing the pro domain to see if that solves the issue is sort of one of the next steps I'm going to try.